So hello students, so welcome again. We are discussing about the chapter 4th. In the previous few part of these chapters we have discussed about the classification systems which were discovered by the Ainsworth and the general characters of fungi. Okay, so now today we will discuss about the life cycle of mushroom, or which is known as Agaricus biosporus. So this Agaricus, it is a sporophytic fungus, we can say, or fungi, which is also called as a Salapta, or it is known as a button mushroom. Okay, so it usually occurs on the dead or the decaying leaves, wood material, or the manure, the agricultural waste, we can say, which is found during the rainy seasons. So they are called as a mushroom fungi. So in India, Agaricus has 17 species and locally known as a we can say kumb or kukurmuta. Okay, so Agaricus is which is known as a common field mushroom, which is also called as a button mushroom. Okay, in the industrial field, or you can say in the commerce field. So its habit, and when we discuss about the habit and habitat one, so this Agaricus, which is sporophyte one, it grows in open field, grassland, and soil rich in cellulose and lignin materials. So on forest floor that is rich in humus formed by decay waste material of plants rich in moisture, so this Agaricus usually grow faster in that area. So it prefers moist shady place, and in rainy seasons the umbrella shaped bodies come out are known as a mushroom. So it comes out with the fruiting bodies, so and that fruiting bodies are known as the basidocarp of the ascospores. So this Agaricus is cultivated mushroom in a well composted manure and at the immature, or we can say the button stages. Okay, so the fruit basidocarp are harvested, which is rich in protein, minerals, salt, amino acids, and fibers. Is supposed to be a ideal diet for the diabetic patients also. So when we discuss about the structure of Thylas, so this Thylas it is called as a mycelium in this. So it remains underground superficial layer of humus rich organic matter. So this mycelium is of two types, that is a primary mycelium and the secondary mycelium. So this primary mycelium is formed by the germination of basidiospores, which is a seed of the fungus. Okay, so this mycelium is a hyaline, septate, and monokaryotic one. So the basidiospores are of positive or negative strain, and thus positive and negative monokaryotic mycelium is formed. So the primary mycelium is short lived, okay, short durations. So it gets transformed into secondary or the dikaryotic mycelium. So the dikaryotic mycelium is formed by the fusion of two primary mycelia of opposite strain, that is a positive and the negative strain. So this secondary mycelium is perennial one, which is branched, and septate one. So it is we can say binucleate, septate, hyphae, and hyphae of such secondary or the dikaryotic mycelia twist together to form a white hypal cords called as a rhizopores one. Okay, so the dikaryotic mycelia form circular colony of hyphae in soil or in the humus. So at maturity, these hyphae develop fruit bodies at their tips, that is known as a basidiocarp one. Okay, so we will discuss about the structure of the basidiocarp. So this basidiocarp is an aerial part of the Agaricus one. So it develops from underground secondary mycelial mat called as a rhizopore one, which is commonly called as a mushroom. So it is differentiated into stalk or stipe and umbrella shaped body called a pilus. So the stipe may be 6 to 9 centimeter in height, while pilus is of 5 to 10 centimeter in diameter. So the pilus has more than 300 to 600 gills at its under surface. Okay, so the gills are radially arranged on the pilus developed on the stem, which is developed on the stem. Okay, so each basidiocarp develop as a small white knot-like structure, so which gets organized into button stage having short basal tip and apical hemispherical pilus with gills formation. Okay, so the button stage get organized into fully grown basidocarp with long strip and umbrella shaped pilus. So the strip has a ring like annulus. Okay, so the strip is a thick, fleshy, cylindrical, light pink or white structure. Pilus has a numerous gills and its inner surface. Okay, so the surface of the gill has a fertile layer called as a hymenium containing basidospores. Okay, so when we discuss about the structure of gills, so in this diagram we can see the tiers of gills. We will discuss about that. The basidocarp is made up of a pseudoparenchymatous mass of hyphae having lower stalk called as a stipe and upper pilus with gills. Okay, so in this the strip or the stipe has a compactly arranged pseudoparenchymatous tissue with peripheral structure, cortex and central region called medulla. So the pilus is also differentiated into an outer cortex and a central medulla, that is called as a gills. So the gills are somewhat complex in internal structure and has three regions, that is a trama, subhymenium and hymenium. Okay, so it is given to three types. So the first one, trama, is a central region of the gill. It has loosely arranged interwoven hyphae one. Okay, in the structure you can identify it. In the next, that is a subhymenium, is called as a hypothecium one. It is present on both sides of central trama. So it is formed by the lateral branches of hyphae of trama. The cells of the hypal branches are somewhat isodiametric pseudoparenchyma. So in this hymenium, next the hymenium is the outermost portion of the gill. So it is a fertile portion of hymenium. The cells are arranged in a palisade like layer. So these are somewhat columnar club shaped cells called as a basidia. So each basidium has 2 to 4 basidospores at the stigmata. So the formation of basidia is the end product of the sexual reproduction of somatogamy. So in this method, hyphae of positive and the negative type form dikaryon by the plasmogamy and the karyogamy, and finally meiosis that gives rise to basidiospores that will be of maybe of positive or the negative one. So this was the structure of gills. Now the next part, that is the reproduction of this agaricus part. So in this agaricus, it is reproduced by vegetative, asexual and the sexual method. When we discuss about the vegetative method, so it is edible mushroom in commerce field and are grown by small piece of dikaryotic mycelium called as inoculum or spawn one. So this when we discuss the asexual reproduction, which is done by the chlamydospore formation. So the chlamydospores are formed on secondary mycelium, which then germinate to produce dikaryotic mycelium. So some species of Agaricus reproduce by Odia formations. So when we discuss about the sexual reproduction in the Agaricus, which is most of the species are heterothallic except in the Agaricus bisporus, which is a homothallic one. So the primary mycelium are formed by the germination of basidospores of opposite stain. So by somatogamy, the hyphae and opposite stain by the dikaryotisation forms the secondary mycelium. So the secondary mycelium get rise to the basidocarpon. Okay. So when you think of the somatogamy, is most advanced method of sexual reproduction in which primary mycelia, that is positive and the negative type, or monokaryotic mycelia of opposite stain by plasmogamy form dikaryotic or the secondary mycelium. Now the karyogamy, where nuclei of opposite type which fuse to form a dikaryon, or we can say diploid nuclei. So it takes place in a young basidium. So after karyogamy, meiosis takes place in the basidium. So the basidospores, that is the positive and the negative, are then formed at the tip of stigmata. Okay, so these are the three types of reproduction and the life cycle of the Agaricus. Now the systematic position of Agaricus. So kingdom is as usual mycota of the fungi, with division is the Eumycota, subdivision is the Basidiomycotina, then class is the Heminomycetes, then order is the Agaricals, family Agaricaceae, genus is the Agaricus. So in the exam, this total life cycle of Agaricus may come. Okay, so you have to start with the habit one until the systematic position. So thank you very much.